the name is a little clunky, but we learned to live with it. It's the disease caused by a virus called SARS-CoV-2. SARS-CoV-2 is the severe acute respiratory syndrome-like coronavirus 2. 2 because there was an original severe acute respiratory syndrome-like coronavirus that was discovered much earlier, in 2002, roughly. So coronaviruses are very familiar to us. They are known because of the kind of crown-like look they have in the electron microscope. These are viruses with an envelope. Those envelopes are decorated with glycoprotein spikes. When you look at them in the electron microscope, it looks like they have a little crown. And corona is the Latin root for crown. In Spanish, it's the same word, corona. And so these are the crowned viruses. There's 4 of them, 4 subtypes of coronaviruses: alpha, beta, gamma, and delta type coronaviruses. Only the alpha and the beta viruses cause diseases in humans. Now, these 4 coronaviruses here that have these rather boring names, 229E, NL63, OC43, blah, 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 these are coronaviruses that just cause upper respiratory infections. In fact, we would say that they cause the common cold. So these 4 have been known for a very long time. These 3 right here are not as well known to us because they're more recent. All 3 of these come from animals. So we call these zoonotic infections. They jumped from animals to humans. So there's the Middle East respiratory syndrome virus, MERS, and that infection, fortunately, never got very far out of the Middle East. There's SARS coronavirus that causes the SARS infection that became prevalent in 2002. It stayed mainly in Asia, didn't really make its way over here. And then more recently, SARS-CoV-2, which has a lot of similarity to the original SARS virus, but causes a disease that's somewhat different. So these 4 coronaviruses that we referred to earlier, these particular viruses, I'm sure each and everybody listening to my voice right now has had an infection from these. They make humans sick with upper respiratory infections. Your nose starts to run, your eyes get red, you sneeze, you cough, you feel awful, and then hopefully in a week, 2 weeks, maybe a week and 2.5 weeks, 3 weeks, whatever, you start to feel better. The 3 more recent ones cause diseases that are much more severe. So common colds, as we call them, are actually caused by a whole plethora of different viruses. There's rhinoviruses, adenoviruses, Coxsackie viruses, and then coronaviruses. All of these cause common colds. So they cause the so-called rhinitis, that's just a runny nose. So-called bronchitis, that's an infection of the upper respiratory tract, the conducting system that leads down into the alveolar sacs of the lungs. And more seriously, in weakened people with weakened immune systems, they can cause pneumonia. If you don't have a weakened immune system, you are not at huge risk from these particular viruses. These 3, however, are a very different story. So SARS caused body aches, chills, and fevers, and then that usually led to pneumonia. And then the Middle East respiratory virus led to shortness of breath, that then led to pneumonia. That surfaced in Saudi Arabia. It continues to be an issue from in that region. This disease was originally reservoired in camels. And because camels, unless you go to the zoo, are not an animal that we have a lot of contact with, this disease never really made a foothold here. Also, this disease likes warm weather. It doesn't like our cold Michigan winters. So it's doubtful, unless this particular virus has some sort of changes it experiences, that this disease is going to find its way to Michigan. SARS-CoV-2, like SARS before it, reservoired in bats. So it jumped from bats to humans. Now, SARS was a problem in Asia. Wasn't much of a problem here in North America. SARS-CoV-2, which causes COVID-19, of course, has caused a pandemic. So this is the one that's giving us so much trouble nowadays, and that's really going to be the subject of today's talk. So here is a electron microscope picture of SARS-CoV-2. You can see it has the nice characteristic crown-like appearance. Now this is a timeline essentially. It's very possible that the virus had been circulating in human populations earlier. But the first recorded case, at least on record, is in late 2019. From there, as you can see, the epidemic moved very, very quickly. There were 27 cases of pneumonia in Wuhan, China. And then you have China announcing the identification of a novel coronavirus as the etiological agent of this unidentified pneumonia. And then a traveler from Wuhan was confirmed in Thailand. That very quickly gave lie to the reports coming out of China that person-to-person contact was probably not possible. Then human-to-human transmission was officially confirmed. There was good evidence before that. Of course, the prevarication of the Chinese government was very well documented. I don't want to get into that here. But WHO then essentially said in late February that we have a problem of international concern. Then they issued a so-called public health emergency of international concern bulletin. And by then, of course, the virus had spread to Europe and then to North America. And by March 11th, WHO said, yep, we have a pandemic on our hands. Wuhan was locked down January 23rd. A week from then, the virus had spread to all 34 provinces across China. And by the end of January, you have a public health emergency of international concern on the earth. And then up here in October, of course, we have over 1 million deaths and then 34 million cases, and so on and so forth. Number of cases just keeps climbing. So what does this virus look like? It has this envelope. This envelope is decorated with these proteins. This is a so-called S protein that's going to bind to the host receptor. The host receptor, the main one at least, is called ACE2. There's another receptor we'll talk about here in just a bit. There's also this M protein that plays a role in the virus getting inside the body. There's a D protein that helps put this whole thing together when it's bloody from the cell. The RNA on the inside is about 30,000 bases in length. It's coated with the so-called N protein. Then there's other proteins involved in the life cycle of the virus. This particular RNA is called a plus RNA. Plus RNA means that when the RNA enters the cell, it can instantly act as a messenger RNA. It doesn't need to be copied into the other sense strand. It can be translated immediately into the protein. That's why it's a plus-stranded RNA virus. So the virus jumps from bats. There might be another reservoir animal. It's thought that pangolins might have also been a reservoir animal. That's debated. The virus has been isolated from bats. So the original strain, the so-called L strain, which was the original strain, that had been found in bats. That jumped to humans. It caused an upper respiratory infection. That then caused a pneumonia. This pneumonia then can be the seeding site for spread to other organs. The ACE2 receptor turns out it is also widely expressed in the GI tract. That's why the virus can cause diarrhea. It's also expressed in kidney, and it's expressed in other tissues, liver, and it's expressed at lower levels in the brain and other tissues as well. It's quite heavily expressed in lungs, and it's quite heavily expressed in blood vessels. That's going to play a role in the pathology of the virus. So the first thing that this virus does is it attaches to these ACE2 receptors. And this spike protein has been very heavily worked on. There's been some very nice electron microscope and X-ray crystallographic pictures of the spike protein. This is a beautiful protein. It's trimeric, which is interesting because a lot of other glycoproteins from lots of other viruses are also trimeric. And when it binds to the target, and I just want to also make reference, this thing has a portion that binds to the H2 target. Then it has other pieces. These pieces actually change quite rapidly. That's where you get different viral strains. These pieces down here are involved in the fusion with the membrane of the virus, with the membrane of the host. These don't change very much. These are probably better targets for drugs than these portions here. This binding, this receptor binding, the target in green. That changes a good deal. It's probably not a good target for drug development. So when it binds to the target, it undergoes a shift from a closed to an open conformation. This shift from a closed to an open conformation essentially drags the virus down to the membrane, and it hits it so close to the membrane that thermodynamically, the most energetically lowest energy thing for it to do is to fuse. And so these helices that, I get my pointer over here for the sake of the people on Zoom. There's the pointer. These helices here, they form this 6-level helix compound here. And that's from this peptide region here and the fusion peptide here. That helps drag the viral envelope next door to the envelope of the host cell, and the thing is now going to fuse. So you can see this very nicely here. It undergoes this conformational change, drags it into here, the fusion of the envelope membrane with the virus membrane. That releases the RNA into the cytoplasm of the cell. The virus also carries its own polymerase, and it encodes its own polymerase. This is a special polymerase. And it's special because most polymerases take DNA and they make more DNA. But they take DNA and they make an RNA copy from the DNA. This takes RNA, makes an RNA copy from an RNA source. So it's an RNA-dependent polymerase. Very unusual. And it will then make copies of this RNA, but before that, you get expression of this, you get a translation of this into the viral proteins. These are then clipped by enzymes called proteases into the active proteins. One goes as polymerase, makes lots of these, makes the viral proteins. Those then assemble in the membrane. The viral proteins then take the RNA and they bud from the membrane and make more virus particles. These then go infect other cells. All right, so if you are younger, if you're say 10 to 50 years of age, usually you'll have a mild disease if you have any symptoms at all. Many people, easily half, maybe even more than half, depending on the age group, don't suffer from any noticeable symptoms. However, if you have symptoms, it's usually very nondescript symptoms. Fever, fatigue, usually a dry cough. The symptom that ends up being seen most of the time is a loss of taste or smell. That is actually diagnostic for this particular virus. Incubation period usually is about 5 days. That can vary, and it varies because different strains, turns out, cause different severities of disease. Also, depends on how much of the virus you got in the first place. If you didn't get very much virus, your chances of getting severe disease are pretty low. If you didn't get much virus at all, you may not get much severe disease, and may not even have symptoms that are really all that bad. Regardless of the case, these early symptoms are pretty nondescript. The virus actually is only replicating in your body for about 2 weeks. After that, the severe disease is actually more due to the immune response. That's usually the case in people who are older, people who have problems with their immune system, or people who have risk factors. In this case, you might be more likely to severe disease. Now, the severe disease is going to occur in other organs typically, and the first organ that usually feels it is the lung. So in the lung, you get infection of the so-called pneumocytes that line the alveoli, and you get this robust inflammation. This little alveolar sac fills with fluid. If it fills with fluid, and then you get fluid also exported out, that fluid will actually start to impinge on this sac and prevent it from expanding. The other thing that happens is you get a mucus plug up here. This mucus plug acts as a one-way valve. So air can come in, but it can't leave. That's called air traffic. And this situation here is called acute respiratory distress syndrome, or ARDS, ARDS. That is a very severe condition, of course. That change cannot occur. This fluid, of course, is due to the inflammation caused by the virus. You have white cells that come in particular T cytotoxic cells. They also recruit neutrophils and other types of white cells. They can do a lot of tissue damage. The vessels here are going to get leaky. That's what causes this to fill up with fluid. That causes this ARDS. The patient may need to be intubated in order to get enough oxygen to their tissues. However, the virus can circulate through the blood, and it can infect these endothelial cells. It infects these endothelial cells, which express a lot of R2 receptors. They can actually cause clots. And a lot of the data that's come back from autopsies of patients with COVID-19 is that there are a tremendous number of clots in the kidneys, in the lungs, in the liver, and also in the heart. Another target for this virus is the heart. And that's essentially what can cause your disseminated disease that's responsible for a lot of your deaths in older patients. This is essentially what the pneumonia looks like in a CAT scan. You get what's called consolidation. Oops, sorry. You have the alveoli filling up with fluid. As they fill up with fluid, they start to look opaque on the CAT scan. This is called consolidation. And then as these areas essentially start to fill up with this inflammatory, actually you start to get what are called ground glass opacities. And then you get things where you can't see through them at all. And then of course the panel F here, as you can see, that's a patient with what's called a pleural effusion. This is very severe, and this patient unfortunately passed away from this disease. If you have a weak stomach, I suggest you not look at the next picture. So 3, 2, 1, you ignore. So here is a lung from a patient who died from COVID-19. You can tell that this lung has cavities in it. And these cavities here are indicative essentially of the inflammation. When you take a section of this, you can see the infiltrate. There's blood in the lung. The vessels are swollen. They're also filled with this inflammatory exudate. That's indicative of ARDS. That's so characteristic of the more severe type of COVID-19. Now, the disease, it turns out, the virus only replicates in the body for about 10 days. This is one of the reasons why that your antivirals only work if you give them early. Because the disease that you have in the first about week to 10 days, that's a result of the viral replication. After this, it's due to the immune system essentially revving up and causing what's called a cytokine storm. This is characterized by immune dysregulation. You essentially have large amounts of these signaling molecules called cytokines that are made. These essentially cause a robust and overwrought immune response that essentially causes massive damage to tissues and also causes something called shock, where your blood vessels essentially massively dilate, you don't get proper circulation to organs, and you undergo organ damage. So this is the reason why these antivirals like remdesivir, these antibodies against the spike protein, things like that, they only work if you give them early, during the viral replication period. After that, you have to give things to calm down this immune response, like steroids and also anticoagulants. Low dose again, anticoagulants have also proven to be very, very effective as well. Your other antivirals only work if you give them early. When a patient comes in and they're severely sick, they're not going to work. A lot of studies that have looked at severely sick patients, they found that these antivirals don't work for this reason. You're essentially closing the barn door after the horses have already escaped. There's a second receptor called neuropilin, and this is expressed in neural cells, and this is the reason why the virus can also divide in brain tissue. And the virus initially probably infects due to the fact that R2 is expressed in the brainstem and neurons at a low level, but then it then can start to exploit neuropilin and infect neuropilin-affected cells, which is virtually all the cells in the brain. This has been shown with in vitro cultures of stem cells, and this is the reason why people suffer from encephalitis and meningitis, very severe disease. Also, people who've recovered from COVID-19 can sometimes have psychiatric manifestations. That's probably due to brain damage due to infection of the brain by the virus. Granted, this is not common, but if you have millions of infections, it's going to be common enough so that the healthcare system needs to be aware of this. So the mortality of this virus by age groups, as you can see, is low if you're young. It's 0.23% overall. Now you might look at 0.23% and laugh and say, I'm in college, I'm an athlete, I'm really healthy. But it depends on your viral load, it depends essentially on the strain you get infected with, and it depends how long the virus lingers over your tissues. So as you start to get into the upper ages, as you can see, your chances of survival go down. And it's different from the flu in that the flu tends to kill a lot of the very young kids. COVID-19 does not. However, the flu and COVID-19 can be deadly to people who are older. Also, if you have pre-existing conditions, this virus, of course, can be deadly. One that I don't have on here is obesity, because obesity will tend to increase your risk for type 2 diabetes, and then cardiovascular disease as well, hypertension, chronic respiratory disease, and then cancer. All of these, of course, increase your risk of COVID-19. Just a word on the present situation. This is from November 9th. This is from the COVID tracker site, and you can see the United States cases are ticking up. The United States is in the pink here. Our cases are ticking up. Of course, we didn't have a very good summer compared to everyone else. You can see Belgium is kind of the basket case amongst the European nations. And mind you, these upticks in Europe, when you acquire them, you don't acquire them in public. Mask wearing is virtually 100% in Europe. Hand sanitizing is virtually 100% in Europe. Where are people getting these infections? They're getting them indoors. When they come indoors, they shut the doors to their domiciles. This infected air now doesn't circulate, and then they invite other infected people over, or non-infected people over. Most of these infections are indoors. And because of that, CDC and the European agencies are now suggesting we ventilate our homes. The uptick in the US is actually quite frightening because this very broad uptick suggests that it might be a spike much higher than our previous spikes as well. Now, just a word on the this Barrington Declaration that came out a few weeks ago. They suggested that we perhaps use the Swedish model as our model for handling this virus. I put this up here. These are deaths per 100,000. In the red, you can see Sweden here. Sweden is actually one of the higher countries when it comes to deaths. So using the Swedish model, here's the UK and the US. The Swedish model doesn't get you very far when it comes to deaths. So the Barrington proposal has been panned by several public health experts simply because Pneumonia treats lockdowns and binary. You either lockdown or you don't. Turns out there's lots of variation between those two. So that's one of the problems. Secondly, if we're expecting herd immunity to do the work for us, it's not going to. Because as we can see, herd immunity didn't really work with the Swedish model. Their deaths are actually higher than the others. And just to show you, Belgium is in fact the basket case. Here's Belgium over here. Why is Belgium such a basket case? Again, most of these infections are indoors. It's cold in Belgium right now, and they close their houses down. They've invited people over. That's where a lot of their infections.
It's colder in Norway. Why is Norway so low?
Why is Norway so low? Because they ventilate their houses, and they don't invite people over. So, what are the recommendations that we should follow, essentially, now that Thanksgiving will be coming? Our motto should be face, space, hands, and air. I know that doesn't rhyme with my others, but hands, face, space, and air. So hands would be hand hygiene, hand washing. I've been very gratified to see students washing their hands very often. That's been very good. Face, don't touch your T-zone. That's your eyes, nose, and your mouth. That's your T-zone. Wearing a mask can prevent that. Also, I've had lots of, I'll put this in quotes, debates with people online about the efficacy of wearing masks. Folks, this is not debated amongst people who study this for a living. Masks not only decrease your tendency to pass the infection, they decrease your tendency to get the infection. This is not debated. Masks are not political. Masks are critical. That's my motto on masks. So, and then also spacing. Meta-analysis studies from the Lancet have shown that masks only work if you combine them with hand hygiene and physical distancing. So the motto there is wear a mask, but treat it like it doesn't work. And then also ventilation. Ventilation. This is critical now because when you're inside your house, you're closing your house up, that air doesn't circulate. CDC has started recommending opening your house for 45 minutes twice a day. So open your windows, go for a walk, come back, close your windows, the job is done. To illustrate the efficacy of masks, Dr. Kunzelman has a nice experiment with CO2, liquid nitrogen, and he's going to show you essentially trying to put the camera essentially where you all can see it. So let me first of all get to where let's see Dr. Kunzelman. I think you can see. Wait a minute. I need to share my all right, so I need to show my camera. Oh, wait a minute. I think I know where it is. Okay, stop that. All right, so you guys should be able to see Dr. Kunzelman there in the corner. Okay? All right, so Tom is going to put some liquid nitrogen into some trays. These are deep trays. They're kind of deep trays, I guess you could say, is that big right now?
They're about, I'm going to say, 3/4 inch deep.
Okay, so the 3/4 inch deep cake trays. So think of a really robust cake, essentially. then he's going to cough or talk over those. Now, the droplets essentially come from your mouth. When they hit nitrogen, they're going to turn into liquid droplets you'll be able to see. And droplets come in lots of different sizes. The virus, very clearly, is passed by droplets. Droplets are about 10 nanometers in diameter. If they're smaller than that, and that number is debated a little bit, if they're smaller than that, they're aerosol. Droplets, if they're small, they evaporate quickly. If they're bigger than that, they will be blocked effectively by a mask. Now Tom's going to show you a cough. Disgusting. Okay. Notice it went off the edge of the table. That's how far the cough down to the mask. And I didn't see a lot of cough there.
You try to breathe?
Now
Breathe.
just do a breath. Okay, yeah. Now do a breath with the mask on.
Gonna try with this mask.
And okay, let's spread. Now, pull your mask down and just talk normally.
What's up, Bradovich?
Now put your mask on.
What's up, Bradovich?
Okay. Now do a little something. Pull your mask down and shout it.
Bradovich! Wow!
Okay? Now put your mask on and shout it. Now, as you can see, the mask blocks droplets. The virus is not passed as a bare particle. It's passed in droplets. And I've had multiple discussions with people online who say that the mask meshwork is far too big to block a virus. That's only true if you pass bare naked viruses. You don't pass bare naked viruses, they're passed in a vehicle. That vehicle is a droplet, and the mask blocks the droplets. Now, other people have said, but I'm not sick. Well, how do you know you're not sick? What if somebody is in the store and they just come back from a chemotherapy treatment? And yes, I know this has happened to me because I walked past someone in the store who was talking to a friend in the store about their chemotherapy treatments. And I not wearing a mask, I would put them at risk. Now, the second thing I'd like Dr. Huntsman to demonstrate is the principle of dilution. The principle of dilution. This is the reason why the CDC is recommending ventilation. Ventilation. This will be important as we celebrate Thanksgiving and we have people coming home. Now, with ventilation, you open your doors, your windows, your whatever, and the air now exchanges from the outside to the inside. If you have virus that's living in your house, and don't forget, this virus has the capacity to survive a while on surfaces. So the virus, for example, can survive 9 hours on skin. That's on your skin. So if you get it on your skin, 9 hours on your skin. It can survive 24 hours on cardboard. It can survive 3 days on countertops, on plastic, stainless steel, and it can survive up to 5 days on wood and glass, and up to 8 hours on latex and aluminum. It is killed by UV light. That's the disinfectant properties of sun. It's also killed by high temperatures, which we're not going to get a lot of during the winter. So Dr. Kunzelman is going to show us, I'm snagged, I can't go any further.
It's okay.
So,
This is, unplug it.
well, but then, then oh wait, but then Zoom. Okay. So,
Alright,
this, and then I go up over here, and
It's not working.
Oh,
I can see it here on the screen here.
okay.
This is basically 50% drain cleaner, and the other 50% water. I'm going to place it into this flask, and if you notice closely, you'll recognize just how concentrated this is when it goes in. You might be able to see. You may have seen like it looked like it got a bit cloudy. That's just an optical effect when you're mixing two solutions that are very different in concentration. But of course, when I swirl it, it mixes thoroughly. Now, every time I take a little bit of this solution and pour it into the next flask, which is just water, I'm diluting 10 times. So I took that 50% solution, I poured it into here, and now this is a 5% solution of the drain cleaner. And every time we do this, I'm going to dilute it by 10 times. There's a 10 times dilution. So that's a total 100 dilution. Try to do this as quick as I can. 1,000 time dilution. Woo! What's it? Help me out. 10,000?
10,000.
10,000.
You can do this on many fingers.
And in 100,000.
100,000 times.
At the end, he's got to drink it.
We go for a cool million.
Going for a million. Just in case. Go all the way out to 10 million.
10 million. So we're diluting at 10 million times.
That's 100.
I think that's 10 million. Let's count together and make sure we got it straight. That was 10 times, 100 times, 1,000 times, 10,000, 100,000, 1 million, 10 million times. So now what I'm going to do is I'm going to add a little bit of an acid-base indicator. Drain cleaner's a base. This particular indicator will turn pink in the presence of the base. A couple drops. So sure enough, the base is in there. This is so concentrated the color may actually fade because the base actually chemically interacts with the indicator, the chemical in the indicator, and destroys it. If this starts to fade, that's what's going on there. The base is so concentrated that it's eating away at the chemical responsible for the pink color.
I'm seeing a lot of pink.
I'm seeing some pink too. I wonder if we're going to have diluted it enough. Uh-oh. I hope so. Uh-oh. Trouble. Trouble. Aha.
No wonder my stuff doesn't work. I mean, your science.
Maybe a faint pink there. You may not be able to see it, but I sure do. Do you see it?
Yeah.
Okay. Just a bit. So this should do the trick. That should be it.
Nothing.
10 million times. Okay.
That was a lot of dilution.
It sure was.
Yeah, but one coronavirus can get you.
Well, this illustrates essentially how we can dilute the virus, and by diluting the virus, let me get my slides all copacetic here. We can ventilate essentially our domiciles, and we can dilute the virus. We dilute the virus, we dilute the infective dose. We dilute the infective dose, we dilute the probability of infection, and we dilute the probability of getting a severe infection. Just by this simple task of ventilating our domiciles. The other feature you'll notice here are HEPA filters. HEPA filters. These HEPA filters can also take the air and purify it. So ventilation in combination with HEPA filters can do wonders, essentially, to making sure our domiciles are free of the virus especially if we have people over. Also, and I say this with some trepidation, at this point in time, with the numbers being so high, you might want to give a second thought to having a lot of people over your home, just because of the risk right now. It might be better to postpone that until later. I'm a parent. I'm looking forward very much to seeing my girls during Thanksgiving. It would tear me to shreds to not see them this Thanksgiving, but if it would be for the sake of their health and my health, and also the sake of the people they interact with, that they might get sick, I think it'd be really worth it. Okay, so those are our recommendations. So hands, face, space, and air. At this time, we would like to conclude our presentation and would like to accept any questions anyone has. Those of you on Zoom, I invite you to type in questions via the chat function, and I will just take questions now. If anyone, these hundreds of people here, have any questions, please go ahead.
The increased risk of problems with diabetes, I assume that was type 2.
Type 2 and type 1.
So they lump them together.
Yeah.
You don't happen to know how those flesh out there.
No.
Okay.
No, the immune dysfunction in diabetes, because of the dependence upon the immune system on sugar, to be able to get those cells to expand, to get those cells to mature, to make cytokines, make antibodies, make the things they need to destroy affected cells. The fact that your sugar metabolism is disrupted means that the fuel for the immune system is also disrupted. So that's why there's an increased risk of immune dysfunction in people with diabetes. That's why they're at higher risk for severe disease. That's a good question.
I got a question. I've heard people say the kind of mask I have doesn't do any good. Could I try it? With
Yeah,
the
sure,
see
you
how
can
it
try it.
compares to what Tom does?
The
Already done
CDC
those
says that face shields help, but they don't help as much as a mask, so they recommend you wear a mask instead of a face shield. If you can wear a mask, if you can't wear a mask because of big concerns or COPD or something like that, then they recommend you wear a shield, because it's better than not wearing anything. So please go ahead and get
Question
it.
about carbon dioxide. Nitrogen up there.
Oh yeah.
I got a question about your
So Dr. White wants to cough on the make this face shield. He wants to see how well the face shield works relative to the mask. And
let's see.
Dr. White over here. Demonstrate. I'll put the camera on Dr. White.
Hey, Ravage! Why? This thing seems to be working.
Yeah, it works pretty well.
Let's see. With the control, that doesn't do any good either.
Get closer.
Aha!
Oh, boy, I can mean it.
1, 2, 3. Seems like this is,
Yeah, it works pretty well.
At
Sometimes
least-
I notice comes down pretty far. Some
yeah.
of them don't come down that far.
Yeah,
Yours
yeah.
does go nice and far.
But it doesn't keep me from getting stuff. It keeps me from infecting. But stuff could still get in here.
Yeah. So, CDC has, their research and the research in other labs have shown that neck gaiters actually don't do a very good job of protecting against either keeping you well or from keeping other people from getting sick. So I see a lot of people on campus wearing neck gaiters. Neck gaiters actually in some cases can get you more sick because they can actually trap things that masks don't trap. Also, masks can be replaced and cloth masks can be washed. Paper ones can be disposed of. Neck gaiters don't tend to be replaced, although maybe some people do. The research on neck gaiters show they don't work nearly as well as masks. So neck gaiters would be choice number 3 over your mask, your face mask and then neck gaiters. Yeah?
How do you classify bandanas then? 'Cause I know a lot of people wear those as well, where they just tie a bandana. Is that more of like a neck
Bandanas? It depends how many layers of cloth you have. The big CDC publication that came out just end of last week essentially said any 2 to 3 ply, the more ply the better. 3 ply is the best, 2 ply is good, 3 ply is better. The bandanas sometimes are just 1 ply. You don't get as good protection. Also keep in mind the mask will block small droplets because of electrostatic charges on the mask. The fewer ply you have on the mask, the less electrostatic blockage you have, you're more likely to leak the smaller droplets. Smaller droplets don't drop to the ground as quickly, they can stay afloat longer, but they'll tend to evaporate more quickly too. So if they get small enough to where they become aerosols can stay afloat for quite a while. Those are the ones that really people worry about. And that's why there was that report, oh gosh, it came out I think 2-3 months ago. Turns out that because the virus can grow in the GI tract, it is shed in sewage, and because toilets create aerosol, there was worry that aerosol from toilets might be a vehicle of transmission. To date, nobody, any reports I've read, has caught the virus from aerosol created by toilets. So that's a possible mode of transmission, but people don't know if that is the case. If it does occur, it's definitely pretty minor. So I wouldn't worry about, battening down the hatches and flush the toilet or something like that. Maybe if you just close the lid and flush it.
Let me jump in as well and answer that. If folks are interested in how this works with other, face guards, thank you. I've got 3 videos that can test a whole bunch of different face guards under a whole bunch of different conditions. Just send me an email and I'll send you the links to the videos. And also, if somebody suggests a test that I haven't thought of, we're certainly happy to run those.
And for Tom's videos, that's Tommy Technicium. That's his YouTube channel. Lots of great stuff there. I can't recommend Tom's YouTube channel highly enough. Great channel. Lots of good stuff, not just for COVID-19, but for science in general. So,
Why are older people?
Yes.
Why are older people why do they die more quickly?
For 3 reasons. Number one, clearance from the upper respiratory tract is not as robust as it is in younger people. 2, the secretory antibody response is not as robust. 3, because the response in the lungs tends to be dysregulated more often in older people than in younger people. So with the dysfunction, you get, instead of the humoral and the cellular response working together, one tends to take off relative to the other. This immune dysregulation is more common in older people than it is in younger people, just because as the immune system ages, it doesn't work as well. Like everything else in your body as you age, unfortunately. That's why older people are more at risk. Also, older people are more likely to have pre-existing conditions that increase their risk. They're more likely to have heart conditions, high blood pressure, diabetes, obesity, things like that. Yeah.
After people wearing multiple masks, is there any research that shows that increases your risk of transmitting or contracting the virus?
Okay, I didn't hear the first part of the question.
People wearing more than 1 mask.
People wearing more than 1 mask. I'm not aware of studies that have addressed that. I will say that in all the studies I'm aware of, the N95 masks without the vents in every study performed the best. So they represent the lowest risk. Whether an N95 is equal to 2 masks, 2 cloth masks, I am unaware of any research on that. It's clear the more plies you have, the more layers of cloth you have in a cloth mask, the more effective it is. I'm guessing if you have a multi-ply cloth mask and you wash it, it takes longer to dry, would be my guess. But it's definitely the most effective. My wife sat down, and she's a person of great sartorial skill she sat down and she made, that means sewing, by the way. That's something she did, and now we have lots of cloth masks, and we wear them, and you can dry them out, and it's really quite convenient. Because it's clear, you're supposed to change, if you wear a paper mask, you should change it probably every 4 to 8 hours. On the airlines, you have to change it every 4 hours. So I just have to plead ignorance and say I don't know. But whether wearing 2 cloth masks is better than wearing 1. Maybe, I don't know, it's not been tested. That's my guess.
I run tests on bandanas with just a bandana in front. It's essentially ineffective. If you fold it in half, it gets a little better. If you fold it in half again, so there's 4 layers there, just a little bit gets through, and you can, check
Right,
it.
so the more layers, the better. And I think that's probably one of the issues with gators, is that the gator only has the 1 layer. You know,
I've done the same with gators as well, doubling up a little bit.
yeah, doubling up the gator works better. That's probably the issue with them, is that they're only just the 1 fly. Okay, yeah, Jenna?
Out of curiosity, is there a specific way you should be cleaning the cloth ones?
The blade should go over your nose and mouth. Beyond that, just make sure that your seal is good across the mask. And just as long as the breathing areas, the 2 ply is over the breathing areas, it should be effective.
I said cleaning, like
Oh,
when you're
cleaning.
washing them.
There's a device that the Chinese developed. It's based on these things that you use for cross-linking gels. When you transfer a gel to nylon, you put them in these irradiators. They adapted those to masks because UV light, particularly UVB light, will kill the virus. If you expose the mask to UVB light for 5 to 10 minutes, it will clean the mask. Also, during the summer, if it gets up to 90 degrees in your car and you leave your mask in the car for 2 hours, the mask is clean. So that's not going to happen, of course, during the winter. But if you were to say, I actually tried this. I tried my toaster oven at home. I tried jacking it up to 350 and leaving the mask there. It made the mask so brittle that it tore, and that wasn't worth it. The cloth masks were much more durable, and they handled it much better. The mask was quite hot when I came out. I had to wait for it to cool down before I could wear it, of course, but that worked pretty well. So heat or UV light washing, if you just wash it with regular laundry soap, or there's a, I don't know if they make this product anymore. They used to make it, it was useful. I was a confirmed bachelor until I was in my 30s. It's a product called Woolite. You used to be able to wash shirts, and do they still make Woolite? They do. Woolite, if you wash it in Woolite, the virus is killed by Woolite. And Woolite is just a wash, hot water, rinse, and hang dry, and they'll kill it. So, soap and water basically will kill it. You should be good to go
So
there.
is it better to use hot water then?
Yeah, better use hot water and soap. Don't just use hot water. Use soap because the virus has the envelope and the detergent disrupts the envelope. That's a good question. Yeah, Carol.
What do you think about quarantine? Like, I know people who have been on the airplane, they'll say they're going to quarantine for like 4 or 5 days. It's like, well, how do you evaluate that?
Yeah, quarantining, where it's been used effectively, quarantining clearly works. The issue with quarantining is that in England and in the United States, we've said things like, well, then self-quarantine, and there's no checkup on people to make sure they're actually quarantining. If you read the CDC report on self-quarantining, it essentially reads like a confirmation of the doctrine of total depravity. People, when they're asked to do something and it's inconvenient, they don't do it. They need to be supervised. Now, does that mean you have the ULO patrol essentially going past their house, and then you put a shock collar on them or something like that? I'm definitely not an advocate for that, nor am I an advocate for the Chinese method of welding people inside their homes. But If you're going to quarantine, then there should be some sort of checkup. Maybe there's a phone app saying, yeah, your quarantine's not over. Have you left your home yet? If you have, tell us so we can contact trace the people you've been in contact with. Quarantining clearly works. Now, if you've quarantined for 5 days and you're negative, and then you're still negative after another 3 days, the chances that you're infectious are extremely low, because it's clear with the RT-PCR test, false positives are not nearly as common as false negatives. Some people, too, are clearly infectious. They've got chronic disease, but they're negative. The reasons for that are not clear. Maybe the virus has undergone mutations and it created a strain that's just not recognized by these tests anymore. Who knows? Also, some people will test positive even after the virus has clearly been driven from their body by the immune system. You can't pick up viral antigen, but the RNA is still there. Nobody knows why that's the case. So your chances of spreading after that are extremely low. I think a 2-week quarantine after those cases are really unwarranted. CDC still says quarantine for 14 days. I think that's overdoing it, but that's just me. So the holy grail really of testing is a home kit. That's cheap and that's effective and it's fast. We're just not there yet with a home kit that you can buy off the shelf and then use to test yourself. Once we get a home kit, I think we'll really be able to not just contact trace, but people will be able to know, am I infectious, am I not infectious? Because you're clearly infectious before you have symptoms. You're most infectious when your symptoms start, but that gray zone, before you start developing symptoms, you're clearly infectious before that time. You're clearly shedding virus. So quarantining does work. Does work. How long should you quarantine? At least a week. At least a week. And if you're negative on day 5 and you quarantine 3 more days, you should be fine. You should be fine.
All right, folks, well, that's 11 o'clock.
Okay. And that's a wrap.
Thank you.
I thank you.